Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and Stem Cell Technologies webinar. I'm Liam Sanyo from the events team at Scientist.com, and I'm pleased to be your host for today's event. We're very fortunate to have Dr. Marta Walasek and Danielle Truong from Stem Cell Technologies here with us, who will be providing tips to lock down effective genome editing conditions for CD34 positive cells, methods to evaluate genome editing efficiency, and optimal culture conditions to maintain stem cell function and long-term editing effects. I'm really looking forward to getting started. And without any further ado, I'd like to introduce the first speaker, Dr. Marta Walasek. Marta, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you, Liam. Welcome to this webinar, in which myself and Danielle will discuss the key steps in designing successful workflows for hematopoietic stem and progenitor cell gene editing, which are the cells and their sources, culture conditions for CD34 cells, and editing parameters. Let's begin with the workflow steps focusing on the cells. Understanding the biology and requirements of a target cell type is critical in order to develop and optimize genome editing workflow, since protocols vary according to the application and cell type. In this part, we'll cover what a hematopoietic stem cell is and how is it different from progenitor cell, as well as why is it important for genome editing workflows. We'll also touch on application of hematopoietic stem cells and their sources. What are hematopoietic stem cells? The stem cell concept was first proposed by Thiel and McCulloch in the 1960s, when the two defining criteria of stem cells were introduced, multipotency and self-renewal. As such, hematopoietic stem cells are defined by the ability of self-renewal and multi-lineage differentiation, as well as can be characterized by the quiescent state, a cell cycle dormancy. These features together allow to sustain HSC pool and hematopoiesis during the entire lifespan of an organism. Hematopoietic progenitor cells are the progeny of hematopoietic stem cells that are generated in the process of differentiation. These lineage-committed progenitors continue through a cascade of differentiation steps, by the end of which mature functional blood cells are generated. Progenitor cells are highly proliferative in contrast to HSCs, have a very limited to no self-renewal potential, and therefore cannot sustain lifelong blood production. HSCs are the only cells within the hematopoietic system that possess this property. Since the discovery of functionally defined HSCs more than 60 years ago, significant progress has been made to identify and purify these cells. Several landmark studies have identified surface markers that allow prospective isolation of functionally diverse stages of HSC development, with a CD34 cell surface marker being readily used to identify HSPCs, a population that contains both stem and progenitor cells. I will use the term HSPC or alternatively CD34 cells throughout the webinar. The CD34 cell population is a heterogeneous population and generally contains very few bona fide HSCs. Therefore, to identify primitive cell, additional markers are required, which are outlined on the graph to the right here. Although an invaluable tool in HSPC purification, cell surface markers measure only the physical properties of the cell, while the most important determinant in stem cell characterization is their functional potential. Up to date, in vivo transplantation remains the only true HSC assay able to assess their hallmark features. Among in vitro assays, colony forming unit or CFU assay is the most commonly used assay for hematopoietic cells. It allows for a relatively quick estimate of the frequency and activity of stem and progenitor cells in a test cell population, and therefore can be used as a method for screening the effects of a treatment, such as gene modifications, on HSPC function and frequency. Hematopoietic cells find application in a variety of areas, from basic research, such as modeling of normal and disease hematopoiesis, drug development or toxicity studies, through research tools in the generation of humanized mouse models to cell therapy. And HSCs are an attractive source for a wide range of cell therapy development, where restoration of blood cells or introduction of normal hematopoiesis is needed, and it offers a curative treatment for an array of diseases such as malignancies, bone marrow failure, immunodeficiencies, and genetic disorders. As such, hematopoietic stem cell transplantations have been successfully used for over 50 years in clinical settings, with more than 50,000 transplants being currently carried out annually across the globe. Therapeutic potential of hematopoietic stem cells can be greatly expanded by advances in gene editing technology, which holds a promise to expand the application of hematopoietic cell therapy to an array of genetic, previously incurable diseases. The spectrum of disorders potentially curable by editing of HSCs is highlighted on the graph here. Additionally, ability to genetically modify patients' own cells offer the same therapeutic benefits as conventional transplants, but without the risk of allogenic settings. Over the last decade, an abundance of preclinical studies have provided proof of concept for the therapeutic potential of gene-edited HSCs. In recent years, the field of gene therapy has been revolutionized by the development of engineered nucleases such as CRISPR-Cas9 by enabling targeting and precise modification of the genome. With CRISPR-based editing approaches being currently a focus of gene therapy research, and over 8 clinical trials performed up to date, mostly focusing on red blood cells disorders so far. One of the attractiveness of hematopoietic stem cells for cell and gene therapy and for ex vivo genome editing is their ease of collecting. HSPCs can be collected from 3 main sources: bone marrow, mobilized peripheral blood, and cord blood. Following collection, CD34 cells are purified from the respective tissues and tested for quality. There are few differences between CD34 cells from various sources. One of them is the number of CD34 cells and recallability of a donor. It has been also shown that in vitro expansion and engraftment potential may vary between cord blood, mobilized blood, and bone marrow, which may have implications for ex vivo culture optimization and definition of cell dose for cell therapy application. These and other considerations need to be taken into account when choosing the source of the cells. But the application and the stage of development will strongly affect this. Currently, mobilized peripheral blood is the main source for autologous transplants and therefore is the most common source of CD34 cells for gene therapy research, whereas cord blood has seen some adaptation in early phase research and workflow optimization. Okay, thank you, Liam. Several discussed features of HSPCs make them an attractive target for ex vivo gene editing, such as the just discussed ease of collection, the capacity to home and engraft upon transplant, and thereby regenerating the entire hematopoietic system of an organism, which is obvious one, and their ability to survive in ex vivo culture, which is the focus on the next section of this webinar, and cell culture is one of the key steps in genome editing workflow optimization. There are few important considerations in defining optimal culture conditions for CD34 cell editing, including how long to culture and why, how to minimize loss of primitive cells, and how to assess whether edited cells remain functional. A successful genome editing workflow should result in gene-corrected HSCs that is capable of generation of lifelong gene-corrected progeny. Therefore, editing strategies that rely on ex vivo cell culture should preserve HSC stemness and at the same time allow for efficient editing. However, primitive cells are challenging to edit and have been shown to be less permissive to genome editing due to their quiescent nature. As well, they tend to be more sensitive to the cytotoxicity correlated with gene editing methods. Pre-activation of cell cycle and exit from quiescence are needed for efficient editing in primitive cells. Though these cells may require longer stimulation compared to progenitor cells, which will be a confounding factor in optimization of culture protocols for heterogeneous samples such as CD34 cells. Additionally, extended culture required for stimulation of primitive HSCs is associated with differentiation and impairment of long-term engraftment capacity. Therefore, optimization of culture protocols for CD34 cell editing and monitoring the frequency of primitive cells in the final sample are critical. Over the last 30+ years, research has focused on defining culture conditions to manipulate the behavior of HSCs ex vivo. Several early studies have shown that HSC survival and activation in culture can be achieved with early acting cytokines as stem cell factor and TPO. However, such conditions and stimulation with growth factors results in gradual differentiation. More recently, several promising strategies have been proposed to partially counteract adverse effects of cell culture. These include identification of signaling pathways and small molecules capable of stimulating self-renewal and delaying differentiation, such as Notch ligands, prostaglandin E2, SR1, and UM171 small molecules. These studies have shown that supplementation of a culture medium with a cocktail of early-acting cytokines plus small molecules helps to maintain the long-term multilineage repopulation capacity of human HSCs. But despite recent advances in ex vivo HSC culture methods and our ability to stimulate self-renewal, cell culture is still associated with a gradual loss of primitive cells. Therefore, the length of ex vivo culture is a balance between HSC activation required for efficient gene editing and the loss of HSCs. With general agreement in the field, the shorter, the better. Several studies demonstrated this in gene editing context, such as studies from Zonari et al., which have shown that phenotypically defined edited HSCs are gradually declining in culture with or without supplementation with SR1. This has a direct impact on engraftment levels and, importantly, on persistence of gene-edited cells in vivo, as shown by studies by Charlesworth et al. And Genovese et al., demonstrating a challenge of decreasing editing frequency and persistence of editing effect when moving up the progenitor hierarchy, and highlighted on the far right graph, but early versus late engraftment potential and its persistence. However, several groups have shown that by tailoring culture conditions, as well as delivery platforms, improvements can be achieved to the yield and frequency of targeted primitive cells. These studies described several critical culture parameters to reproducibly achieve high editing frequencies in CD34 cells. Cell density has been shown to affect editing frequencies, with lower density cultures resulting in higher expansion rates and increased editing efficiency, and these two parameters has shown a positive correlation with each other. Another parameter shown to play a key role in optimization of editing workflow is the length of pre-editing culture. As discussed earlier, the quiescent nature of HSCs may require longer stimulation time. And along these lines, extended culture protocol before cell editing has been shown by Genovese et al. To increase significantly the percentage of edited cells in primitive population, which translated to the increased percentage of edited long-term engrafted cells in vivo. And 48 to 72 hour stimulation before editing has been shown by multiple groups as optimal to achieve cell activation and increase editing efficiencies. Last but not least, The length of post-editing culture plays an important role in optimization of gene editing workflow. This culture length should allow for assessment of editing efficiency and functional effects of editing while preserving primitive HSCs. A shorter ex vivo culture is preferred, and as shown by the findings from Genovis et al., is associated with higher in vivo engraftment potential when comparing 4-day versus 12-day post-editing culture. Importantly, this study also showed that the combination of all of the key parameters discussed here, low density culture, optimal stimulation length, and minimal post-editing culture, together with the use of small molecules, was able to extend rare gene-edited primitive HSCs during tested protocol. And keeping in mind the discussed key parameters, now I will walk you through the study we performed to optimize and validate the culture conditions for genome editing workflow for CD34 cells. The same workflow was used across all experiments, including medium and supplement testing, as well as optimization of genome editing. As a source of HSPCs, we use human cord blood, from which CD34 positive cells were isolated. Freshly isolated or thaw-frozen CD34 cells were then plated in low-density cultures for 48 hours to allow for stem cell activation prior to genome editing. Next, the cells underwent genome editing using CRISPR-Cas9 system. Design and optimization of the editing parameters will be discussed by Danielle in the next section. Following editing, the cells were cultured at low density again for additional 4 days to allow readout of culture conditions on editing efficiency and HSPC number and function. By the end of this 6-day protocol, effects of tested workflow were evaluated using both physical assays such as immunophenotype as well as by functional assays using CFU assay. We evaluated 3 different serum-free hematopoietic medium: StemSpan, SFEM2, XF, and AOF. The difference between the media is the level of compliance from serum-free through xeno-free to animal-free. The media were tested in combination with 3 different expansion supplements: CC100, CC110, and CD34 expansion supplement, which differ in regards to early-acting cytokines. Additionally, supplementation with small molecule UM171 was evaluated to promote optimal maintenance of HSCs during ex vivo culture. First, performance of the media and supplements was evaluated in regards to cell viability and editing efficiencies. Performance of each medium was tested across various expansion supplements as well as in the presence or in the absence of UM171 small molecule. Each medium type is grouped in its respective group and color, with SFEM2 shown in orange, AOF in gray, and XF in green. Supplements are shown on the graph in following order: CD34 expansion supplement first, CC100 second, and CC110 third. The addition of UM171 molecule to the respective cytokine supplement is indicated by striped bars. Overall, the data shows that all of the tested culture conditions performed similarly across the board and support high cell viability, as shown on the left plot, as well as high editing efficiency demonstrated by the right plot. Next, the effect of various culture conditions were tested in regards to cell expansion and frequency of CD34 cells. Focusing first on the orange-colored part of the graph to compare effects of various supplements within one medium type, we can see that the choice of cytokine supplement affects both cell number, shown in the left plot, and the frequencies of CD34 cells, shown on the right plot. Among tested supplements, CD34 expansion supplement stimulated the best cell expansion and frequencies of CD34 cells. Of note here is that cell number is notably lower compared to what's expected in expansion protocols, since this workflow specifies optimal conditions for CD34 cell editing, not expansion. Maintenance of CD34 cell frequencies was further improved by addition of UM171 molecule depicted by the striped bars on the right plot, irrespective of cytokine supplement, and resulted in optimal CD34 cell numbers and frequencies. The similar trends were observed across other tested medium, StemSpan AOF and StemSpan XF. And based on these data, we concluded that all media, SFEM2, AOF, and XF supported CD34 cell culture in genome editing workflow, and that among cytokine supplement, CD34 expansion supplement in combination with UM171 molecule supported optimal CD34 cell maintenance. Next, we focus on the identified optimal condition and characterized it in more detail. The graph here highlights that the combination of CD34 expansion supplement and UM171 maintain high frequency of CD34 cells across tested StemSpan media, as shown on the graph on the left, and importantly, these conditions support expansion of genome-edited CD34 cells, as shown on the graph on the right. We also tested the performance of other commercially available media, which is shown in the dotted bars here. Since optimal genome editing protocol for CD34 cells should allow efficient editing of primitive cells, we looked at frequencies of immunophenotypically defined HSCs following the 6-day protocol. As shown by the data on this slide, we can see that both the frequency on the middle plot and the numbers of primitive HSCs on the left plot were maintained in these cultures. Importantly, high proportion of primitive HSCs were effectively genome edited, as shown on the plot on the far right, showing proportion of edited and non-edited cells within the primitive population. Similarly to other parameters, all tested StemSpan media were able to maintain phenotypically defined HSCs and overall supported effective genome editing in this primitive population. The dotted bars here once again show performance of commercially available alternatives. In addition to immunophenotype analysis, we performed CFU assays to assess the effect of the optimized editing workflow on the numbers and frequency of functional progenitor cells. As shown on the schematic on the left, the cells after 6-day genome editing protocol were plated in metacooled medium and cultured for 14 days, followed by colony counting and classification. The colony counts indicate that gene-edited HSPCs retain their colony-forming potential, as shown by the middle graph, and cell plating efficiencies, as well as colony distribution, shown in the far right graph, in regards to percentage of cells giving rise to erythroid, myeloid, and mixed colonies. And finally, to demonstrate the importance of maintenance and targeting of primitive HSCs in genome editing protocols, we sorted various populations of CD34 cells representing their differentiation status and plated them in CFU assay. 4 cell populations were sorted: CD34 low cells representing more differentiated cells, CD34 mid population, and CD34 bright population containing HSPCs. From CD34 bright population, we sorted additionally on CD90 positive and CD45 array negative cells representing the primitive stem cell population. When plated into CFU assay, it is very clear, as shown by the images of the CFU assay or the far right graph showing plating efficiencies, when the majority of a colony forming potential is contained within CD34 bright population, and more specifically is derived from primitive HSCs that also showed the highest CFU frequencies. Importantly, the vast majority of the most primitive colony type, multilineage CFU GEMM colonies, were contained in the primitive population, highlighting the importance of primitive cell maintenance during editing protocols. To briefly summarize this part of the webinar, we discussed and showed that cells itself and cell culture are key in gene editing workflows, where healthy and active cells are required for editing, where a requisite of successful and reproducible cell culture are standardized tools and optimized protocols, and that fine-tuning of culture conditions and timing are crucial to promote effective editing while preserving long-term persistence of engineered cells.
Danielle, it's all yours.
Thank you, Liam and Marta. In this section, I will talk about genome editing of CD34 cells using RCTech CRISPR-Cas9 system. First, I will introduce to you CRISPR-Cas9 system. Then I will discuss the typical experimental workflow, focusing on the 3Ds of CRISPR, the design, delivery, and detection. And finally, I will discuss some results of CD45 and B2M knockout experiments. Genome editing involves the deletion, insertion, or modification of the DNA sequence at a specific location in the genome. Prior to discovery and application of CRISPR-Cas system, genome editing was accomplished using engineered non-specific nucleases fused to sequence-specific DNA binding domain of protein. The DNA binding domain are designed to provide target specificity, and the nucleases function to introduce double-stranded DNA breaks. The CRISPR-Cas system differs from these traditional methods in terms of nuclease targeting. Targeting of the Cas effector nuclease is based on Wash-and-Crick base pairing between a guide RNA and the target DNA. All genome editing relies on repair of nuclease-dependent DNA breaks by endogenous DNA repair mechanism, which I will discuss in more detail in the next few slides. The use of CRISPR-Cas9 as a genome editing tool was first described in 2013, where it immediately propelled genome editing into mainstream conversation and widespread use by scientific researchers. The simplicity of guide RNA design for new target, the need for only a single constant protein, and the modest cost and complexity of this platform have made targeted genome modification readily accessible to scientists around the world. The NET technology has enabled detailed study of gene function and variant of unknown significance, disease modeling, and gene therapy. In the commonly used CRISPR-Cas9 system, Cas9 enzyme from Streptococcus pyogenes forms a complex with a guide RNA, which directs Cas9 enzyme to a specific DNA target sequence. The guide RNA is made up of transactivating CRISPR RNA, sometimes known as tracer RNA, which bind to the Cas9 enzyme, and a CRISPR RNA, which base pair with the target sequence. The target sequence must be adjacent to a short sequence motif, known as a proto-spacer adjacent motif, or PAM, in order to be bound and processed by the Cas9 guide RNA complex. With the target sequence identified, the Cas9 enzyme makes a double-stranded break at the target site. The break will be repaired by the cell via an endogenous DNA repair mechanism. Non-homologous end joining, or NHEJ, is the predominant DNA repair pathway in mammalian cells. So if the cell chooses to engage in this pathway after the DNA break, there are two primary outcomes. The sequence can return to wild type and be free of errors, or insertion and deletion, or sometimes known as indel, can form if the DNA ends are processed prior to ligation. I will present two examples of CD45 and B2M knockout that are the result of the NHEJ pathway in the later section. Homology-directed repair is the second major DNA repair pathway. It has the ability to faithfully copy the genetic information from a related DNA sequence or an exogenous foreign DNA sequence. HDR function in late S phase and G2 phase. However, most cells spend the majority of their time in the G1 phase. Therefore, this pathway occurs with a low frequency in most cells. In the context of CRISPR-Cas9, this pathway enables precise rewriting of the genome if an exogenous DNA template is added to the experimental design. There are some challenges in genome editing of CD34 hematopoietic stem cells. As Marta mentioned previously, hematopoietic stem cells are a challenging cell type to culture and manipulate in vitro due to the cell lose stemness over time. Also, several reports have recently shown that homology repair is more efficient in progenitor cells as compared to LT-HSCs. Although current investigation attempt to increase homology repair rate in LT-HSC, this is still the biggest hurdle in accelerating clinical strategy for homology repair-based genome editing for blood and immune system disorders. In this section, I would like to, however, discuss more about the issue in expression and delivery of CRISPR component into CD34 cells and the editing off-target effects. Earlier attempt to apply CRISPR-Cas9 for genome editing in CD34 cells using mRNA-based method activate the innate immune system of the host cells. Other methods such as plasmid-based system, on the other hand, resulted in low targeting efficiency and high toxicity in CD34 cells. There are also some reports showing chemically modified single-guide RNA may be more efficient at editing HSPC. These expression systems also pose concern about safety in clinical translation due to the risk of unwanted genetic mutation and immunogenicity. There are a few different methods to express CRISPR-Cas9 component in cells. This is critical as CRISPR-Cas system doesn't naturally occur in mammalian cells. This image may help visualize the mode of CRISPR-Cas9 expression and delivery system. There are typically three methods: DNA plasmid, mRNA-based, and RNP system. For the CD34 workflow, we utilize the RNP system, which represents the most efficient method of delivery. First, there is no molecular cloning required. Unlike DNA plasmid, which requires both transcription and translation step, and the mRNA-based system, which requires the translation step once the system delivers the CRISPR component into the cells. RNP system bypass all these steps and can immediately enter the nucleus for editing. Also in RNP, gRNA is protected by Cas9. Therefore, it is less prone to degradation compared to mRNA-based system that include naked mRNA, which is not very stable in cells. The RNP system is also the safest method of delivery, since there is no chance of plasmid integration. When modified guide RNA are used within the RNP system, it also does not activate the cellular immune response as we often see in IVT mRNA. And finally, since Cas9 protein is degraded fairly rapidly, the opportunity for Cas9 to generate DNA break at affinity of target site is reduced. In this study, Nguyen et al. Demonstrated that across a number of different electroporation conditions, RNP or protein-based delivery of Cas9 outperformed both DNA and mRNA in terms of indel generation as shown in Figure A. Figure B is a Western blot analysis of Cas9 expression after being delivered into the cell using DNA plasmid, mRNA-based, and protein-based method after 0 hours and up to 72 hours post-delivery. When CRISPR-Cas9 is expressed on a DNA plasmid, it does not show expression until approximately 8 hours post-delivery and continue to exhibit very high expression level, even at 72 hours after delivery. mRNA expression of Cas9 begin rapidly after delivery. However, the expression level is quite low. In contrast, protein-based or RNP delivery show high expression after 4 hours and up to 24 hours. It is reduced by 48 hours and completely absent at 72 hours post-delivery. In Figure C, taken together with off-target analysis at two predicted off-target sites, for this guide RNA, there is a clear relationship between prolonged expression and increased off-target effect, with DNA-based expression showing the highest level of off-target effect compared to mRNA and protein system. We have developed a fully integrated workflow for high-efficiency genome editing of CD34 cells. This includes resources to help with gRNA design, instruction for preparation and delivery of RNP-based Architect CRISPR-Cas9 system into CD34 cells, media to support CD34 pre- and post-editing, and analysis method to detect the editing efficiency. In this section, I will focus on some consideration for gRNA design, method of delivery, including viral and non-viral methods, and a few methods of detection of your edited samples. So gRNA format can impact editing efficiency. As shown here, gRNA can be produced or expressed in 3 different formats. Two-part or single gRNA can be purchased commercially as synthetic modified gRNA. We utilize this modified single gRNA format in the RNP system in the CD34 workflow. Alternatively, single gRNA can be transcribed in vitro and purified in the lab or encoded on an expression plasmid. However, studies have shown that synthetic modified gRNA are still the best and the more flexible in terms of editing efficiency across different complex cell types. It requires few preparation steps prior to delivery and exhibit higher editing efficiency compared to IVT or plasmid expressed gRNA. The additional modification to the end of synthetic gRNA is also critical, as discussed earlier, as unmodified gRNA can be degraded by intracellular nucleases and can activate a cellular immune response. So how to design gRNA? The first step is to identify the target reference sequence by looking up the target gene sequence using a genome browser such as UCSC. For example, for the B2M knockout in the CD34 experiment, I will look up the B2M gene sequence and focus on early exon. In this case, we target exon 1 of this gene to ensure the more efficient knockout. Then one should design PCR primer, flanking the sites of interest and perform sanguine sequencing of DNA. This step is optional, but it is highly recommended to ensure no SNP in the target sequence. Some of the most popular gRNA design tools include CRISPR, which is shown here. Once you have your target sequence, you can input a sequence into the gRNA design program. The design algorithm then scans the input sequence for potential CRISPR RNA as 20 nucleotides followed by the PAM site. So it's also scanned the target genome for similar sequences for possible off-target that matches to each CRISPR RNA. gRNA design programs are useful, but gRNA activity is highly recommended to be tested in the lab, since the frequency of NGG PAM site is approximately 5.2% in the human genome. Most genes will have multiple potential CRISPR RNA target sequences. Typically, at least 3 independent CRISPR RNA are tested in parallel to derive distinct edited population in order to independently verify functional effect of the mutation. There are quite a few number of non-viral methods to deliver cargo into cells, which include physical and chemical methods. Electroporation is one of the physical methods. That is a technique that applies a strong electrical field onto a cell membrane. The field temporarily reorganizes the membrane, making it more porous and allowing large biomolecules to pass through. This delivery method generally uses higher editing efficiency. However, high cell death may result from high voltage pulses and incomplete membrane repair. Therefore, the electroporation parameter must be finely tuned for individual cell type to avoid reduced long-term cell viability. Chemical delivery also utilize the formulation of complementary molecules to assist Cas9 to bypass the cellular barrier, as well as to protect it from degradation pathway. Some chemical transfection methods include polymer chemistry, lipid-based, and nanoparticles. Chemical delivery typically use lower editing efficiency compared to physical and viral method. Transfection efficiency often depends on cell type as well. Most common cell line and immortalized cell line are generally easy to transfect. However, primary cells and stem cells, on the other hand, are more sensitive and often have lower viability after transfection. Viral methods also give high editing efficiency. Lentiviruses are effective in a variety of cell lines, primary cells and stem cells, and can be used in vivo. However, the biggest concern of lentivirus is that because they integrate randomly into the genome, they may disrupt vital gene or alter regulation of gene expression. Adeno-associated virus, or AAV, is another type of virus commonly used in vivo editing. AAV typically do not integrate into the genome and will not cause disruption of genes through insertional mutagenesis. One drawback of AAV, however, is the physical limit to the amount of DNA that can be packed inside the virus, since it has the packaging capacity of only 4.7 kilobase. Therefore, designing a virus to encode such a large gene such as Cas9 along with gRNA using AAV is challenging. Here I will briefly describe 4 popular strategies for screening edited cells. The first strategy is based on a mismatch cleavage, also known as T7 endonuclease 1 assay. In addition to this technique, Sanger sequencing or flow cytometry analysis of cell surface receptor or protein can also be used to assess editing efficiency in poor cell population. Targeted or unbiased next-generation sequencing approaches can also be used to assess both on and off target editing efficiency with very high sensitivity. However, this technique can be very costly for optimization. For the CD34 editing experiments, we utilize both flow cytometry analysis for cell surface receptor detection and the T7 endonuclease 1 assay, which I will discuss here. This assay utilizes a T7 enzyme that has a single-strand cleavage activity specific to heteroduplex structure. In the context of a gene editing experiment, genomic DNA of a population of edited cells will be amplified by PCR surrounding the target site. If a non-homologous end joining repair has introduced a mutation, denaturing and annealing will form a fraction of heteroduplex of mutant and wild-type PCR amplicons. T7 endonuclease 1 will recognize this loop and cleave the DNA mismatch in those heteroduplex loops. The relative proportion of uncut and cut fragments provide an estimate of genome editing within the population. Overall, this technique is a useful tool to quickly screen for the presence of indel. However, it is not informative for the exact kind of indel or change. In this section, I will present some results of CD45 and B2M knockout of CD34 cells. The single guide RNA sequences can be designed using stem cell CRISPR design tool once a target locus for editing is identified. We use the same cell source and cell culture condition that Marta described previously, and editing efficiency was analyzed using RCTech T7 endonuclease 1 kit or by flow cytometry. In the CD45 knockout experiment, we first optimized electroporation condition at a few different parameters. So first CD34 cells were cultured in optimal culture media for 4 days and electroporated at 3 different electroporation settings. Cells were then analyzed by flow cytometry 3 days post-delivery. The data shown in Figure A is the post-electroporation recovery that was calculated by dividing the total number of cells collected at 3 days post-electroporation by the number of initial electroporated cells. Figure B shows cell viability that was measured by PI dye exclusion. And in figure C, CD45 editing efficiency were assessed for non-electroporated control cells that electroporated with Cas9 only, and RNP electroporated cells. Overall, the electroporation condition at 1600 voltage is the most optimal in terms of recovery, viability, and editing efficiency. Here are more results of CD45 knockout experiment. Figure A shows a representative histogram of CD45 flow cytometry from RNP electroporated and non-electroporated cells, shown in orange and gray color respectively. Figure B and C show editing efficiency and cell viability that were monitored by flow cytometry using a fluorophore conjugated CD45 antibody and 780 dye occlusion respectively at 4 days after electroporation with either the neon transfection system or nuclear factor system. So the result here showed that RNP electroporated sample exhibited loss of CD45 expression on the cell surface, which show here as about greater than 60% average knockout efficiency, while maintaining viability at greater than 80% average viability across all tested donors. Figure D showed the result of the same tested donor that exhibited loss of CD45 expression. However, the percenter of CD34 expression cells were unaffected by RNP delivery. And in figure E, post-electroporation cell recovery was negatively impacted by the delivery of RNP complexes containing CD45 gRNA compared to the non-electroporated sample. This is consistent with previous report of delayed HSPC proliferation in response to genome editing with programmable nucleases, including CRISPR-Cas9. We also perform T7 endonuclease 1 assay to confirm indel generation from the CD45 knockout experiments. He showed genome editing or cleavage efficiency of 4 donors at 4 days post-electroporation with the neon transfection system was shown in figure F, or the nuclear factor system was shown in figure G. Depending on donor, you see here the cleavage efficiency were confirmed that it detected around from 44 to 70% by T7 assay. So next, we targeted a second locus of CD34 cells to validate the optimized protocol. We chose to target B2M, which encodes the accessory chain of MSC class 1 molecules using a single CRISPR RNA sequence, targeting exon 1 of B2M. So CD34 cells were electroporated with RNP complexes using the neon transfection system. The results here are similar to the CD45 experiment, with the RNP electroporatic sample exhibited loss of MSC1 expression, as shown in figure A and B, while the percentage of CD34 expression cells were unaffected by RNP delivery, as shown in figure C. Here is also confirmed in figure D that both RNP and Cas9 only electroporated cells exhibited reduced post-electroporation cell recovery relatively to non-electroporated control, suggesting that electroporation is the primary contributor to reduce CD34 cell recovery and growth in the genome editing workflow. We tested our final protocol, which include edited cell culture in StemSpan media and a few other commercial medium. Using Architect CRISPR-Cas9 system, B2M effectively editing efficiency as shown in figure A was comparable across medium condition. However, StemSpan SFAM2 media showed the most optimal maintenance of CD34 expression, as shown in figure B, and also expansion of more primitive subset of HSPC phenotype after genome editing, as shown in figure 4C. In figure C. So this indicate that StemSpan media provide an optimal media system to support CRISPR-Cas9 genome editing in CD34 cell. Yep. Thank you, Liam. So in summary, there are a few key considerations for genome editing in CD34 cells. First, cell source, cell culture, and editing parameters are key considerations for optimal genome editing workflow. The CRISPR-Cas9 RNP system is the most suitable currently available method for CRISPR-Cas9 genome editing of primary cell um, because they are the more efficient and has lower chance of off-target effect compared to other expression and delivery system. And finally, careful consideration of pre and post editing culture condition is required to obtain high editing efficiency and to maintain downstream cell function viability and the CD34 cell stemness. We would like to acknowledge the people who are involved in genome editing and molecular tools and hematopoietic stem cell R&D who were involved heavily in developing the CD34 genome editing protocol. And with this, we conclude this webinar, and thanks to everyone for joining us. And we are available to answer any question that you may have for us.
All right, fantastic. Thanks so much, Danielle and Marta, for the great presentations. And as a reminder, if you do have a question you'd like to ask, you can submit it using the Ask a Question panel shaking on the screen there. All right, so Marta and Danielle, ready to answer a few questions?
Yes,
Sure.
we are.
Fantastic. All right, we'll start off with a great question here. What are the potential side effects of using gene therapy, using gene editing?
Okay, I can answer this. So gene therapy using gene editing has some potential side effect. As most gene therapy currently deliver edit gene using a vector, this technique presents some risk. One of the risks is unwanted immune system reaction. The body's immune system may recognize the new delivery vehicle, and this may cause inflammation. Um, Another risk is that the virus carrying the edited gene can target the wrong cell. Since virus can affect more than one type of cells, it can also infect additional cells or healthy cells, not just the one that intended for repair. There could be another possibility of causing a tumor if the new gene get inserted in a wrong target in your DNA. So in regard to SSPC, careful assessment of nuclease specificity and off-target is mandatory when aiming for clinical translation of engineered SSPC, because these cells will have to support lifelong hematopoiesis by performing several cycles of cell renewal and differentiation in the patient. Therefore, any adverse effect from off-target need to be carefully evaluated.
Yeah, definitely. That's a good point. Definitely a powerful technique, but important to control the risk. All right, next question here. How many cells do you typically obtain from a single CBU using the EasySet method? And how quickly do they double within the first 2 to 3 days? In my hands, I've been getting slow doubling over the first few days, and then the cells appear to take off, which I feel is not how they should grow. Thanks a lot.
Yeah, I can answer this question. So with EasySet method from usual core blood unit, it a little bit will depend if method of isolation is positive EasySep or negative. But with positive method on average, and it varies, of course, on the quality and the unit, it's around 1 million of CD34 positive cells that you can get. And the next part of the question is how quickly do they double? This will as well depend if the cells were previously frozen or used fresh. But usually what we observe that during first 2-3 days, there is not much proliferation happening when you look on the cultures as a bulk. And then, yes, indeed, they start taking off around day 4, which is very usual to this culture system. But it's because we are looking at them as a bulk culture. If you plate the cells at a single cell level and you will trace these cells individually over every day on day 0, 1, 2, 3, 4, 5, what you will notice that not all the cells will proliferate. There will be some cells that start proliferating already after 24 hours, but some cells which will start proliferating only after 48, 72 hours, and this will be correlated where in the hierarchy of CD34 they are more primitive or more progenitor. So the reason why we don't see proliferation in first 2-3 days in bulk cultures is because there's in the background going a lot of kinetics on a single cell level of some cells already proliferating while others not.
Perfect. Yeah, great.
I hope that answers the question.
Yeah, I think so. So in your experience, what approach in vivo versus ex vivo would be most suitable for correcting a single base mutation in sickle cell disease in human subjects?
I will take this question. So there are two therapies, obviously. So in the in vivo gene therapy, new gene are inserted directly into your body, while with the ex vivo approach, it's involved removing cells from the body, modifying them, and then placing them back into the body. So between these two approaches, the ex vivo approach is often used in blood-related disorders, such as in this case sickle cell disease, because it utilizes a person's own cell renewal stem cells, which can be engineered with a functional knockout, functional working gene, and then delivered into your body. So as we know that the sickle cell disease is caused by mutation of a single base in the DNA sequence of the beta globin gene, it is a perfect candidate for CRISPR-mediated gene therapy. Currently, many clinical and preclinical development, including treating sickle cell anemia with CRISPR, involve this ex vivo procedure where hematopoietic stem cells are extracted from the patient, corrected them through a homology-directed repair to knock in the normal sequence of the gene, or other strategy involve a gene knockout by switching off the gene that suppress the fetal hemoglobin, which is a healthy hemoglobin, which in turn cause the fetal hemoglobin to be expressed, and in turn, replace the mutated adult hemoglobin. So yes, the answer is ex vivo is more favored for this type of treatment.
Excellent. All right. Yeah, another great answer there. I think in the interest of time, we'll just try to squeeze one more question in here. How can you make sure that the cells are engrafted after re-injection into patients, especially because cells were expanded in vitro?
I can take that. So the dose of CD34 cells is known to influence an outcome of stem cell transplantation when we are talking about fresh cells. And of course, number of functional CD34 cells has been shown to be predictive of engraftment potential, and this can be measured. The frequency of functional CD34 cells can be measured with CFU assay, which in terms as well has been shown to correlate with engraftment potential of non-cultured cells. After culture, of course, cell culture, dependent on conditions of the cell culture that we are using, they will change the frequency of functional engraftable stem cells. And therefore, it is very important to carefully assess the functional potential and frequency of functional stem cells that are remaining in the population of the cells after cell culture, and this can be done using functional assays such, for example, CFU assay.
Excellent. All right, perfect. Thanks, Marta. Yeah, thanks so much, both of you, for the really great presentations, as well as your insights in the Q&A. It's been a real pleasure having you with us.
Thank you. Thank you, everyone, for listening.
Yeah, definitely. Big thanks to the audience for participating. And last but not least, of course, we'd like to thank the sponsor, Stem Cell Technologies. So we now do invite you to take a moment to please fill out the survey that we have here. We do read all your comments and really appreciate any feedback you can provide. If you still have a question, it's not too late. You can still submit it using the Ask a Question panel there, and we'll forward it along to the speakers following the event. And finally, if you'd like to learn more about the research, we invite you to check out the resources panel here, where we've added a few links that you can check out. So in closing, we hope you enjoyed this scientist.com webinar, and we'll see you again next time. Have a great day, everyone.